আমাকে দেখা যাচ্ছে?
Good morning, Rajat sir.
Good morning.
Good morning. Dear students, good morning to everybody. From the behalf of Department of Microbiology and Biotechnology, I am welcoming you all to the second day of our introductory course seminar or webinar. Today the talk will be delivered by our head of the department, Professor Rajat Pal, and the topic of the talk is biomolecules, the molecules behind the biology. But before starting the webinar, I want to share something that is very much painful for us. We have lost one of the greatest leader of India, our former president, Professor uh, Pranab Mukherjee. So in this context, we all want to pay some respect to this person, Pranab Mukherjee, and we want to have 1 minute silence in the respect of Pranab Mukherjee. Thank you, everybody. Again, good morning, Rajat sir.
Yes, good morning, sir.
Professor Rajat Pal has completed his BSc and MSc in microbiology from Calcutta University. Thereafter, he has carried his doctoral work at Calcutta University from famous Rajabajar Science College campus, and thereafter for the last 10 years he is in this profession, in this teaching profession. He is a very beloved professor to all of the students, and I personally know him very well. He was my senior throughout my career, throughout the We had a long journey together for many days, and hopefully, you will enjoy her talk very much. And uh, it is very much good to see that we have almost 35 participants in this live class today, and in the online platform also we have already 20 participants. So it will increase with time. We still have 7 minutes left. So from 10 o'clock sharp it will start. But before that, if Professor Pal wants to interact with you, I want to give him some time, so that he is always very much happy to interact with the students. So I think before starting the talk, he will interact with you for 2 to 4 minutes. Uh, so over to you, Professor Rajat.
Thank you very much, Professor Thakur Chaudhuri. It's an immense pleasure for me to be introduced by you. And I must say that to all the participants that it's a very kind nature of Professor Thakur Chaudhuri that he always tell anyone else that they are very beloved teacher for the students. But the fact is that Professor Thakur Chaudhuri is the most beloved teacher of our department. So all the students are actually always telling about him. Okay, so we are just behind him under his guidance, how to manage the students and everything. Although by age I'm a little bit of senior than him, for that reason I have been chosen for heading the department. But by ability and popularity, Professor Thakur Chaudhuri is far, far, far away than me. Okay, so my beloved students and dear participants throughout the world, I'm very much happy that I've come to know that many international participants has also joined our program. This is the first program what we are actually doing in such a large scale. So many participants are there. Almost 450 students have been registered. So we are for the first time tackling a huge crowd. So maybe sometimes there may be some difficulties we are facing or some technical problems are going on. So for that reason, I beg a pardon to all the participants. Please forgive us and bear with us, be with us for the next 4 days. It was a 5 day program. Yesterday was the talk by Professor Calcutta. Today I am going to talk. And one announcement, little bit announcement, I just want to share with you that another speaker, Dr. Sanjay Malik, he supposed to deliver his lecture today also, but unfortunately for his busy schedule, we have to reschedule his lecture, and he will be, talking on 4th in the last day of the this webinar series. With he will be sharing the screen with Dr. Milan Bonik. So it is almost 10. I think maximum of the students have already joined. And I just want to tell few words about my department. The participants maybe who has not joined yesterday, some new participants may join. For them, I am just telling that our department is a new department, is a baby department. It has been established in 2017. It's a 3 years old department, and it's a very big pleasure and immense, opportunity for me that I was the first faculty appointed in this department. And after that, I have four very eminent scientists with me as a faculty in my department. That is Professor Thakur Chaudhary, Professor Fatima Calcutta, Professor Milan Bunik, and Professor Atri Ghosh. I hope all of them are present today here, and they will support me throughout this lecture. And the most important thing is that in our university, in the very beginning, Professor Ashok Ranjan Thakur was the founder vice chancellor of this university, and he was from our department. He actually established himself this department, and after him, Professor Dhrubajyoti Chattopadhyay came, and he is now the present vice chancellor of our university, and we are very much fortunate enough that he is also belongs to our department. So these two eminent scientists, these two pillars are with us So that is a very big opportunity for us. So with the guidance of these two, eminent scientists, we are trying to run our department very smoothly. Let's see how much we can be succeed. If all of you support us, then definitely I am very much sure that we can succeed. So it's almost 10 in my clock. So may I start? Professor Calcutta, are you there? Hello, Professor Thakur Chaudhary or Professor
Yeah.
Calcutta? Yeah. Hmm.
Yes, sir, I think it is 9:59 in my clock
Okay.
also, so we
Hmm.
can start our
Yes.
session today.
Hmm.
And as we can all see that we have already we have good number of participants and it is increasing with time. So
Hmm. Hmm.
hopefully more will join with us. And best of luck to my Rajatda. Okay,
Oh,
bye.
thank you. I need your good luck. And Dr. Calcutta, is she joined? She was actually trying to call me, but as I'm in this
Okay, I am
webinar, so
I am calling.
okay, you just contact with her if she's, willing to say something. I'm starting. Is this visible?
Yes,
Is this visible?
yes, it is visible.
Okay, okay. Thank you. So, good morning to all of you once again, and I'm starting my presentation for today. Yesterday, as you all see, have an experience for the journey through the life of a cell from birth to death. So today, what we are going to talk about is the biomolecules. Professor Calcutta told you that how a cell can born and how it can journey its life, how it can pass through its life journey, and at the end it dies. So throughout his life, actually it needs from molecules. Okay, these molecules are known as biomolecules. So that we are going to talk about today. What are the biomolecules? Actually, these are the molecules, normal, simple molecules. Okay, in chemistry, molecules are the combination of more than one atoms. Sometimes a very small molecule, in case of very small molecule, one single atom can use as a molecule, but in biology, as it is a complex system, here one single atom cannot act as a biomolecule. So for being a biomolecule, we need more atoms to form a biomolecule. First, it is a molecule, and as it is a very important factor for the biological system, for that reason, we are calling these molecules as a biomolecule. And these molecules are actually doing everything in our living system. So I am Rajat Pal, Assistant Professor and Head of the Department of Microbiology and Biotechnology, Sustainability University. With you all are going to start the journey of the biomolecule. So in my first slide, I'm telling that what is a biomolecule? Biomolecule, also known as biological molecules that are produced by cells and living organisms. Biomolecules have a wide range of sizes and structures and perform a vast array of functions. Four actually major biomolecules are present in our body. What are they? Lipids, nucleic acids, carbohydrates, and proteins. These four macromolecules, in the meantime, I just want to share with you that these biomolecules are also known as macromolecules, biological macromolecules, as they are large, quite large in size. For that reason, these are known as the macromolecules, not micro. Whenever I will tell about the structures, you can understand that how big molecules they are. So they are also known as the macromolecules. So among these macromolecules, I mean, four types of macromolecules are there. One is the lipid, nucleic acid, carbohydrates, and proteins. These four macromolecules are deputed for different four activities in the cell. Not in the cell, in the whole universe. If you consider, then only two things can be happen. One is the work, what we are doing, and number two, the energy behind the work. Okay, without the energy, we cannot do the work, or the work, whatever we are doing, everything is the work done. So the work and the energy behind the work, these two things are the most important thing in all over the universe. And as the cell, our cell is the miniature form of our all the universe. So they also must have these two, things. That means they have to have some work, and they need some energy to do that work. So these four macromolecules are actually deputed in different roles to perform these whole phenomenon. That means the work and the energy production behind the work. First, I'm telling carbohydrates. Okay, carbohydrates is the main component for the production of energy. Okay, as I told you that energy production is must for doing the work done. So carbohydrate is the first element or molecule, the most important molecule which are responsible for the energy production in the cell. Now carbohydrate actually used or is being utilized for the production of energy, that's fine. But what about the lipid? Lipid are the storage of the energy. As always, we have two places for keeping our money. Money is our currency. Whatever we are willing to have or willing to buy, we spend money. So there are two places where we can, keep our money as safe. One is our wallet, our purse, our money bag. There we used to keep our money for daily uses, and for, further use, or for, huge amount of money, we generally use our bank account. There we store the money. From the bank account, we generally don't take the money regularly, right? From there, we take the money and we keep that money in our wallet, and from the wallet, we usually use it regularly. So here also, in case of cell, they actually keeps the energy for regular use within the carbohydrate. And for the storage purpose, they are using lipid molecules for longer use. Okay. So the wallet or the money bag for the cell is carbohydrate, and the bank is the lipid molecules. Okay. So these two are clear, I think, for you, that the function is being performed by carbohydrates and what function is being performed by the lipid molecules. Then, as I told you, that everything is all about the work. What for we are having this energy for the work done, right? So whenever we are getting the energy from either carbohydrate or the lipid molecules, they should be used for the work done. Now, who will do the work? Protein molecules will do the work. In our cell, all the work done is done by the protein. There are several types of proteins present in our cell, whether it is an enzyme, whether it is an antibody, whether it is a membrane protein, transporter protein, whatever it is, everything is done by a protein molecule. So protein molecules are actually the worker in our cell. Okay. And the production of energy by carbohydrate or lipid, and to being use those energies for the work done by protein, all these phenomena are controlled by nucleic acid. Whether nucleic acid is the brain of the cell, he is the, head of the, like we have a head of our family, either our father or our mother or grandfather, grandmother, any one of us, elder person, he is the head of our family. Like that, nucleic acid is the head of the cell. Okay, he actually instructs what to do and when to do and where to do. Okay, so nucleic acid is actually supporting or taking care about the series or sequence of the work done, whether it is going on properly or not. So these four macromolecules or biomolecules are present in the cell, and they have different kinds of activities in the cell. Now moving on to the next slide, this is the types of biomolecules. Here you can see that what I told: carbohydrates, lipids, proteins, and nucleic acid. acids. These four are the major parts of the types of biomolecules. And among the carbohydrates, actually carbohydrates are the sugar molecules. There are many types of sugar molecules are there, what we are going to discuss later on. In the lipid, there are fats or oils. Fats are mainly composed of fatty acids and glycerol. Then the proteins. Proteins are composed of amino acids, and amino acids are bound together with the peptide bond. Nucleic acid, there are two types of major nucleic acids are present. One is the DNA, the other is the RNA. Deoxyribonucleic acid and ribonucleic acid. Okay, so these four are the macromolecules, and these are the components of these four biomolecules. Next, in the next slide, I will tell something about a very beautiful element in the periodic table, that is the carbon. Okay, I don't know why, maybe for some reason, carbon in his previous birth, he has done some tremendously good job. For that reason, maybe God has selected or nature has selected it as the important part of the whole universe. In all the periodic table elements, carbon has been chosen to be the backbone of all the biomolecules. Okay? Without carbon, nothing is there in the universe. Now we are going to see some special properties of carbon. The most important thing is that carbon has a valency of 4 and it has a tetrahedral structure. Okay, what does it mean by tetrahedral structure? The tetrahedral, hedron, the term tetrahedral has come from the word hedron. Hedron means the plane. Okay, so carbon has actually 4 different planes for its 4 valencies, and the planes are arranged in such a way that never ever 3 bonds will be in a same plane. That means 2 bonds will be in a same plane, one bond will be above the plane and the other bond will be below the plane. So likewise, the carbon atom is arranged in a 3D space. Okay, and it has got the duty to be in a central charge of all the molecules. Whenever we will see, you will see that carbon has to be present in all the biomolecules. Okay, so now move on to the next slide. We are going to start the carbohydrates. What are the carbohydrates? Carbohydrates mainly what we are, it is known to us as a sugar molecule. Carbohydrates are a group of naturally occurring carbonyl compounds that also contain several hydroxyl groups. Hydroxyl means OH groups. Okay, it may also include their diversities, which produces such compounds on hydrolysis. It actually derivatives. Sorry, may have some polymer by synthesizing the combination of more than monomers, one types of monomers, and by the hydrolysis of that polymer or oligomers, the monomer can be synthesized. Okay, and the carbohydrates which are soluble in water. One second, please. Yes, sorry for the interruption. Um, the carbohydrates, which are soluble in water and sweet in taste, are called as sugars. As I told you, that most commonly we are used to talk about the sugar molecules, and these sugar molecules are the carbohydrate molecules in nature. Okay, I am showing in the figure. Is it clearly visible? Now I think it will be better, right? STC sir, is it better now?
Yes, sir, it is now better. Now
Yes,
more
yes, yes.
visible. Okay.
Yeah, more visible. Yes, yes. So here actually 3 types of sugar molecules we are showing here. Sugar molecule can have 3 carbon or 5 carbon or 6 carbon. I'm showing that 4 carbon sugar molecules are also there in our biochemistry or macromolecule. But here, the most popular or common sugar molecules I have shown, there's the glyceraldehyde, ribose sugar. This ribose and deoxyribose are, found in DNA and RNA. When I will talk about the DNA and RNA, you can see that these sugars are very much important for their structure. 6 carbon sugars are also there, like glucose, fructose, and galactose, which are very, widely available and used in our daily life. Okay. So in the next slide, here, as I told, that we are showing the types of the carbohydrates. Carbohydrates may be, broadly 4 types: monosaccharide, disaccharide, oligosaccharide, and polysaccharide. In some cases, we actually club this disaccharide and oligosaccharide together, but monosaccharide and polysaccharides are totally separate groups. In case of monosaccharide, what happens? Mono, it means one, single. Saccharon and saccharine means the sugar unit. So monosaccharide means it's composed of one unit of the sugar molecule. Okay. The example is glucose, fructose, or galactose, anything. I mean, whether it is composed of only one molecule, one monomer of the sugar, then it is known as the monosaccharide. Disaccharide, when the molecules are composed or the sugar molecule is composed of two different monomers or monosaccharides by using some glycosidic bonds, then those are known as the disaccharide sugar molecules. As for example, maltose and lactose or sucrose, these are the disaccharide molecules. So as I told you that oligosaccharide and disaccharide sometimes may be clubbed together. That oligosaccharide means 2 to 10 sugar molecules. So disaccharide may fall under the roof of the oligosaccharide, but I have just differentiated between them. Oligosaccharide means if 2 to 10 monomers or monosaccharide molecules are bonded together to form one sugar, that is known as the oligosaccharide. Okay, raffinose, sacchiones, these are the um, examples of oligosaccharide molecules. Next is the polysaccharide. As you all know, that polymer means it's a whole N. That means any number of units. Okay, that can be any number, but larger than the 10. Hmm. So these are, if we have, say, for example, 20 or 25 repetitive units of the monosaccharide or any other sugar, one or two types of sugar molecules, then it's known as the polysaccharide molecules, as for example, starch, glycogen, and cellulose. So these are the major four types of the carbohydrate molecules. Now we are going to start about the monosaccharides. As I told you that in monosaccharide only one molecule of sugar will be present. So simple sugar of carbohydrate and often called simple sugars since they cannot be further hydrolyzed. If we hydrolyze these sugar molecule, then we can have OH groups, carbons and all those things, but we cannot have any other sugar. For that reason, these are the smallest unit of the sugar molecule. Okay. These are usually colorless, is a crystalline solid which are soluble in water and ionizable in a non-polar solvent. Okay, I hope that you all have the clear idea about the polar and non-polar solvents. So if we solubilize these sugar molecules in a non-polar solvent, then they may have some ionic dissolution, a charge distribution amongst them. These are the compounds which possesses a free aldehyde or ketone groups. Okay, the sugars, these monosaccharide sugars may have two different types of groups. One is the aldehyde group, the other one is the keto group. The sugar which contains the aldehyde as their functional group, they are known as the aldoses, and the sugar molecules which are actually which does have ketone group as the functional group, they are known as the ketose molecules. So two types of sugar molecules depending on their function. One is the aldose, the other one is the ketose. Here you can see that glucose and galactose, they are the aldose sugars and fructose is a ketose sugar. Okay. And their general formula, every one of you I think know that CnH2ON or CnH2nON, these kind of things. The examples are very much well known to all of you that glucose, fructose, dextrose, ribose, ribulose, everything is a example of the monosaccharide molecule. Now the disaccharide. When two different sugar molecules get together to make a bond between them to form a dimer of the sugar molecule, then this is known as the disaccharide. Okay. As an example, here I am showing that alpha glucose and fructose, they are actually these two, OH groups are clubbing together to release one water molecule to make a glycosidic bond over here to form a sucrose molecule. Okay. So sucrose is the dimer composed of monomer of a glucose and a monomer of a fructose. Okay, so this is a very nice example of disaccharide with the sucrose, lactose, maltose. These are also the disaccharide molecules. Next is the polysaccharide. In polysaccharide, same unit or different unit units will be repeatedly interacted or bonded to form a polymer with a unit of n. N means it can be any whole number. It can be 20, it can be 25, it can be 30 or more than that. Okay. So when these kind of monomers are bound together to form a long chain, then it is known as the polysaccharide molecule. They can be of two types. One is the homopolysaccharide, the other one is the heteropolysaccharide. What are they? In case of homopolysaccharide, the single unit or the same unit will be repeated one and one and one and one. Okay. If the same unit is repeatedly be added to form the polymer, then it is known as the homopolysaccharide. And if more than one, I think two or more than that, different types of sugar molecules are get together to form a single polymer, then it is known as the heteropolysaccharide. From the name, you can see that homopolysaccharide and heteropolysaccharide, homo means the single unit, I mean similar type of things, and hetero means the different types of things, heterogeneity, and different types of things will be there. So in case of heteropolysaccharide, different sugar molecules will be present, and in case of homopolymer or homopolysaccharide, a single type